Hi friends, let us talk about genetic engineering today. Is also called recombinant DNA technology because it deals with the construction of recombinant DNA. What is a recombinant DNA? Recombinant DNA is basically combination of two different DNA. So this is a vector DNA, a plasmid DNA, a vector DNA, and when it is recombined with another DNA, say this one, then what results is a recombinant DNA. This is a recombinant DNA. So all those techniques that are involved in making this recombinant DNA will be together called recombinant DNA technology or genetic engineering. Genetic engineering or recombinant DNA technology involves creation of new individuals with those are called genetically modified organisms or genetically modified microorganisms. These are also called transgenic organisms. And the process is also called transgenesis. So the techniques involved in making these genetically modified organisms will be together called genetic engineering also. But it is not that always we need to have a genetically modified organism for the sake of genetic engineering. There are targets. The purposes may be to know the function of the gene, characterization of the gene, sometimes gene editing, all these things which are involved in characterizing and knowing the function of the gene or storing it or making transcript of the gene, and then seeing the function of the transcript and its location in the cell. All these molecular biological techniques can be together called genetic engineering. But in true sense, genetic engineering involves engineering as such with the gene, with the DNA element. When you cut a DNA element and then you ligate it with another vector or DNA element, then it is, in true sense, it is genetic engineering. Otherwise, the whole subject is called the molecular biology. So genetic engineering involves a number of steps. Those steps can be enumerated as follows. The first of all, we need to have insert DNA, that is this part of the element, this part of the recombinant DNA. You need to have that first, and it is a particular gene of interest that's need to be transferred, gene of interest. So you need to isolate first the gene of interest. Then the next thing you have to select a vector DNA. Vector DNA is a vehicle. It is usually a plasmid or a patch DNA, or it may be a combination of the two like cosmids, plasmids, and many more types of vectors have been designed nowadays. The next thing you need to have is a number of enzymes. The most important among them are restriction endonucleases, by which you make cleavage in the DNA, both the insert DNA and the vector DNA, for the cloning purpose. Then you need to have ligases with which you ligate the two DNA elements, and then you need different types of modifying enzymes that might involve DNA polymerase, RNA polymerase, clinophragments, and then polynucleotide kinase, simple client phosphatase. Many different types of modifying enzymes are required for genetic engineering purpose. That will be discussed in some other class. So we were enumerating the steps involved in genetic engineering. So first of all, that we saw that you require insert DNA or gene of interest to be isolated, then vector DNA is required to be selected, that it may be a plasmid DNA or fast DNA, and many other different types of vectors are there. Any one of them might be required depending on the purpose. Enzymes of different types are required. Most important among them are restriction endonuclease, ligases, and different types of modifying enzymes. The next thing that will be required is a host cell within which you have to transfer the DNA. Basically, it is E. coli DNA at the initial stage, in which the particular, if it is an E. coli cell, particular recombinant DNA that is being made is transferred to this E. coli cell, just for the sake of storing, and then it is kept it in either in liquid nitrogen or minus 70 degree centigrade deep freezer for future use. And another thing that you require is electrophoresis instrumentation instruments, just for the sake of monitoring the changes that you are making, just to monitor that the recombinant DNA has been synthesized, it has been transferred to a host cell, and that is the recombinant DNA. You can take out this DNA and you can check by making the same restriction digestion, and then you can run on a gel to see whether the same band is being formed or not. So to verify or to monitor all these changes that you are going to make during the time of recombinant DNA technology, you need electrophoresis instrument also. So we will now see in our next class different types of enzymes that are required. Let me give you a brief idea about that. So enzymes that are required during genetic engineering, you may classify them into two types. One is called modifying enzymes, another is restriction endonuclease. So restriction endonucleases are a type of enzyme, and these are basically endonuclease, that is, they introduce cuts inside the DNA, but the name restriction is there. That means these DNA are provided, these enzymes have the capability to restrict the entry of foreign DNA. Restriction endonucleases are usually present in many bacteria, just to protect their cell from foreign invaders like bacteriophages. A bacteriophage, when it delivers its DNA inside a E. coli or any other bacterial cell, it might replicate inside it, or it might be degraded or aborted. Most of the cases it is aborted because the bacterium, it possesses a sort of enzyme, those are called restriction endonucleases, that cleave this DNA and do not allow them to replicate inside. But many a time the phage DNA is successful in replicating inside by a mechanism called modification. So all this restriction endonuclease system has two components. One is restriction, another is modification. The modification is methylation. So this system is together called restriction modification system. A restriction modification system is present in a bacterial cell, that with that system it either allow the DNA to replicate inside, the foreign DNA to replicate inside and complete its life cycle, and when it does, then modification phenomena function is being taking place. And if it does not allow the foreign DNA to replicate, then the restriction part of it is active. So both the system are together present, that is called restriction modification system. It might allow the cell, the phage DNA to replicate, or it might restrict also at the same time. So the restriction modification system involves it controls whether a particular bacteriophage can infect a host cell or not. If this host cell is, suppose it is a E. coli host cell strain C, within this cell lambda C phage can replicate and lyse the cell. It is the host cell for lambda C. So if it is a host cell for lambda C, then lambda C phage, the DNA of lambda C phage will not be restricted. It will go modification, or it has already gone modification in its DNA. With this same modification facility, the bacterial cell also protects its own DNA. The modification is basically methylation. The different places in the DNA are methylated, so that these are the sites where the restriction endonuclease will introduce cut. So if those sites are methylated, the site is not at all a substrate for the restriction endonuclease now. But if this lambda C phage infects another host of E. coli strain K, suppose, then in the strain K, this lambda C phage will not be able to survive. Its DNA will be restricted. It will be clipped into fragments by the restriction endonuclease system. So the whole system together is called restriction modification system. If out of 10 to the power 4 phages, one phage survives or forms plaques, then this phage had probably gone modification. It has gone methylation, its DNA, and so its DNA is getting protected. If this phage further reinfects the strain K, it will completely lyse the cell, and 100% plaque forming efficiency will be seen. So this is what restriction modification system is about. There are 3 types of restriction modification system. Those are called type 1, type 2, and type 3 systems. Among these 3 types, type 2 is most important, because this type 2 restriction endonuclease system is specific, means it introduces cuts at its original cleavage site, whereas these are non-specific. This is also non-specific. It introduces cuts about 1000 base pairs away from the original cutting site, and type 3 also it incorporates cuts about 25-26 base pairs downstream of the original cutting site. Because these 2 enzymes have a cognate methylase activity, they have their methylase activity attached to the enzyme. That's why the site where they need to be cut, suppose this is the site where this enzyme has to cut, it methylates the site. While going to cut, it immediately methylates it, so the enzyme cannot sit here, it slips down to some other place and introduces cuts elsewhere, about 1000 base pair on either on the upstream side or on the downstream side. Same is the case with this enzyme also. It also methylates the original site. But type 2 restriction endonucleases have a different methylase activity that is not attached with the restriction activity. So when restriction is taking place, no methylation is taking place. So it always introduces cuts at a specific site, making a specific ends or termini that can be ligated to another termini of similar compatible type. Type 2 restriction endonucleases, they require magnesium ion and ATP. But this type 1 and type 3, in addition to their requirement of magnesium ion and ATP, they also require S-adenosylmethionine for their activity. For today's purpose, let us end here. Then we will later see that there is a subversion of type 2, it is called type 2S type. So we will describe this type 2 and type 2S type in our next class. These are